So, dear students, in last time we discussed about the gene regulation in prokaryotes, and at that time there was a little discussion about the lac operon, and today we are going to discuss about the lac operon in detail. But before that, what is an operon? Some basic concepts should be there. A clear understanding of these concepts should be there so that the confusion may not take place. So, what is an operon? As operon is a term used particularly in case of bacteria. So, operon, the term indicates that is an operational unit. So, what happens in case of microorganisms, bacteria, that suppose these are three genes Z, Y, and A. These three genes are required for a particular function, that is digestion of a particular metabolite such as lactose. So here, to use the lactose is a sugar disaccharide molecule just like glucose. So to use lactose as an energy molecule, this bacterial cell has to produce certain enzymes, and these enzymes are produced by genes present on the bacterial chromosome. So if that gene is, first gene is Z, so it produces suppose an enzyme and it is represented suppose A. This enzyme has a particular function on lactose. When this enzyme acts on lactose, and suppose this lactose is converted into another molecule C and D, now the enzyme product of this Y gene acts on these products, and similarly the A gene product acts on the other products produced by the action of this Y gene enzyme. So here, and ultimately the bacterial cell can use this lactose in that form that is possible to be utilized. So here, the production of enzyme production by these three genes is necessary to catalyze a particular metabolic reaction. So these three genes lie one by one, first Z, then after Y, and then after A gene. So these three genes are not separated in space, these are attached together parallel one by one. So these three or more than three gene, set of these three, we are taking example of lac operon, and in that three genes are used. So these three genes are regulated by a single regulating unit. So that regulating unit is here, this is known as P, that is promoter. We know promoter is that sequence around messenger RNA or DNA where a particular protein binds, and then uh, whether it starts its replication or protein synthesis or messenger RNA production, that particular sequence where an enzyme or a protein binds, that is called here promoter. So we are looking at this promoter site, and if these are the structural genes and their protein product is necessary, so first these genes must have to be transcribed, and for transcription RNA polymerase must have to bind at the promoter site. So the promoter comes, bind at the promoter site, RNA polymerase comes and bind at promoter site, and then transcribe these three genes, and we get a messenger RNA of these three genes. So here, this single molecule of messenger RNA has the information transcribed for the protein production of lac Z gene, Y gene, A gene. Suppose this much portion of this messenger RNA is used for the protein production of this gene. This much portion is used for the protein production of this second gene, and this rest part is for this third gene. So here only one messenger RNA is produced for all these three structural genes. So this type of messenger RNA is known as polycistronic RNA. Cistronic RNA. When a messenger RNA molecule has the coding sequences or information to produce more than one type of protein, then that messenger RNA is known as polycistronic messenger RNA, and this is the characteristics of microorganisms. So here we are moving back. These three genes are regulated together in group by a common regulating structure. So this structure forms an operon, that is a cluster of gene. Those are regulated in together, not separated but in together. Their product is required in together but not separate. So this is an operon, and we have an example of lag operon. So here we take the example of lag operon. And what is this lag operon? As the lag stands for lactose. Lactose is a disaccharide molecule, just like carbohydrate other molecules, and this lactose is used by microorganisms only when the glucose is absent. That is, glucose is absent and lactose is present. When glucose is absent in the bacterial growth medium and lactose is present, then bacteria use lactose as their energy molecule, is an energy substrate. So the question is that if glucose is present but lactose is absent, definitely glucose will be preferred as a energy molecule. And if lactose is present, glucose is also present, in further in that case glucose will be used as energy molecule. And lactose can only be used as energy molecule when this glucose is completely exhausted, when it remains no more. But the question is, and as last time we discussed, that the lag operon is a negative inducible operon. So here a general structure of a lag operon we can have. These are, suppose, DNA sequences. This is regulator gene represented by a. Now here this much sequence starting from here to here, this is promoter, and this much sequence that coincides, that overlap the promoter sequence, is the operator sequence. And now these are the structural genes, lag Z, lag Y, and here lag A. These three genes are there. So this complete assembly of various regulating units, structural genes, is known as an operon. So here, if glucose is present and lactose is also present, so first focus on lactose. When lactose is present but let's take glucose is absent, as we have already discussed, that glucose will be used as an energy molecule, and in this type of lag operon, that is lactose operon, this lag or lactose molecule act as an inducer. So how? Because this regulator gene I produces a protein, and these proteins are known as repressor. So when the lactose is absent, this repressor protein will bind at the operator sequence. Repressor protein will bind at the operator sequence and thus inhibit the attachment of RNA polymerase. RNA polymerase, another enzyme that binds on the promoter to transcribe these three genes. If the repressor molecule is bound at the operator, it is not possible, or it causes a difficulty for RNA polymerase to bind at the promoter sequence, and thus the transcription and further translation of these three structural genes or the production, enzyme production of these three genes do not take place. So here it is the role played by the repressor in absence of lactose. So when lactose is present and glucose is absent completely, now this microorganism uses lactose as energy molecule. How? Here the lactose molecule at an inducer, and this lactose molecule reacts with the repressor molecule and changes its conformation, changes its three-dimensional structure, and this lactose repressor, or here this inducer repressor complex, is not or unable to bind at the operator sequence, and that's why operator they can't bind at the operator sequences, and RNA polymerase come and bind at the promoter, and then it starts transcription of LAC Z, LAC Y and LAC A gene. The protein product of these three genes is important, but LAC Z gene that produces beta galactosidase, beta galactosidase, and LAC-Y beta galactosidase permease, and A gene beta galactosidase transacetylase. But the actual biological role of this beta galactosidase transacetylase enzyme, or the protein or the function of this LAC-A gene, that is the last gene of this operon, is not still known. So here the question for you, that if presence of, when lactose is present, the lac operon starts. When lactose is present with glucose, then lac operon remains switched off. So how it is, what is the mechanism that in presence of glucose and even in presence of lactose that act as an inducer molecule in absence of glucose, so when lactose is present with glucose, the lac operon cannot run. So what is the role played by this glucose in making it switch off condition, in switch off, to make it this lac operon in switch off condition? So here this is actually cyclic AMP plus CAP protein complex. Cyclic AMP, CAP. CAP is the catabolite activator protein. This protein facilitate the RNA polymerase to bind at the promoter. Even the regulatory gene product, that is repressor, is present, then this catabolite activator protein makes a combination or a complex with cyclic AMP molecule, and then this complex molecule binds at this particular zone of this promoter. So here this is AMP CAP complex binding site just after the regulator gene. So the concentration of cyclic AMP depends on the concentration of glucose. When glucose is present in sufficient quantity, the cellular concentration of cyclic AMP goes down. it reduces, it decreases. And if cyclic AMP is not sufficiently present, then catabolite activator protein, this CAP protein cannot bind or cannot have a complex with cyclic AMP, and if this activator protein cannot bind at at its respective site on promoter, then it no one is there to facilitate the attachment of RNA polymerase over this promoter sequence, and hence there is no production of protein product of these three structural genes. So the glucose, presence of glucose affects the concentration of cyclic AMP. If cyclic AMP is sufficiently available, then CAP protein forms a complex, and it makes the lag operon in on condition. It starts the transcription of these structural genes, and then production of their enzymes product. So, and when glucose is sufficiently present, the concentration of cyclic AMP goes down, and that's why in presence of glucose the lag operon remains switched off. So now let's begin with these lag operon. So here first we have to prepare it in good condition so that it may be in easily understandable and visible condition. So here we represent, this is the DNA sequence, and these are, this is the sequence for regulator gene I. This is the promoter sequence. Sorry, again this is too much short. This is promoter sequence. Oh, sorry. This is a DNA sequence. Suppose this is a regulator sequence I, this is promoter sequence P, and this is an operator sequence O, and these are lag Z, lag Y, and lag AG. These three, this is the general arrangement of a lag operon. So, what are the products of lag Z gene, lag Y gene, and lag A gene? So, lag Z gene produces beta galactosidase. Lag Y gene produces beta galactosidase permease, and lag A beta galactosidase transacetylase. The biological role of this gene or this protein is not clearly known, so we will focus mainly on lag Z and lag Y gene. So now, and what is the role? What is the function of beta galactosidase? It breaks the, suppose this is a lactose molecular disaccharide molecule. So this is glycosidic bond, and this galactosidase breaks this bond and converts this lactose into glucose and galactose molecule, and then this glucose molecule is used in energy. This beta galactosidase not only converts lactose into galactose and glucose, but it can convert this lactose into allolactose, that is an effector molecule for this lag operon. And lag Y gene, it produces an enzyme beta galactosidase permease. So here you can remember it with the name permease. It means it functions, suppose this is a bacterial cell and here lactose present in its surrounding medium. So before this lactose can be utilized for metabolism, this lactose should be transported inside the bacterial cell. So this beta galactosidase enzyme transport this lactose inside the cell, and then inside the bacterial cell the other protein acts on the lactose, and then metabolism of lactose takes place. But one more question for you, that if still this lac operon is in switched off condition and there is only lactose remaining in the medium, surrounding medium, and the bacteria has the opportunity to utilize the lactose, only lactose, but to utilize the lactose it should be transported in the cell, and its transportation is carried out by beta galactosidase permease, that is the product of lac Y gene. So if lac operon is already switched off, then there is no production of beta galactosidase permease, then and if the transportation of lactose does not take place inside the cell, then how the lac operon will start? This is a very basic problematic question. So the answer is that lac operon is a somewhat complicated process. It is not so simple as we will and as we are describing it. In a bacterial cell, when there is lactose present or absent, listen carefully, when it is present or absent, emitter in the background, that is inside the bacterial cell, a slow rate of transcription of these genes always takes place. That is, a minimum level of these enzymes are always present, because if this regulator gene produces repressor molecule and that repressor molecule binds at operator, so technically RNA polymerase cannot bind over this promoter because it is adapted by the or occupied by the regulator gene product, that is repressor molecule, and till the repressor molecule is attached on operator there is no possibility of transcription of these genes, and if there will be no transcription there will be no production of these enzymes. But what happens, sometime this protein, this regulator protein, that is the repressor molecule that is attached on this operator molecules may fall down, may fall down, and whenever this molecule fall down in the bacterial cell, RNA polymerase come, bind and transcribe these genes into their product. So this is the reason that even if lactose is absent, there is a minimum concentration of availability of these enzymes in the bacterial cell. Now, one by one, there are some more terminology, but that will come. So, as we know, a bacterial cell is haploid. It has only one copy of each gene, unlike the human beings and many higher plants where there is two copies of one gene or two alleles of or more than two alleles of one gene. So in bacterial cell, that is haploid, and each gene has one copy, that is one copy of lac Z gene, lac Y gene and lac A gene. So first we take the various combinations of mutations in lac genes. So what happens if lac I gene is mutated, lac P gene is normally functioning, lac O gene is mutated. So there may be a number of combinations of mutations or functioning gene. So it is it will be quite interesting for you to listen it carefully, to watch it. So first we take the representation of these lag gene. So here the genotype of bacteria, and here the product of lag Z gene, and here the product of lag A gene, and similarly here the product of lag Z and lag Y gene. And when inducer is absent and when inducer is present, you know that in presence of inducer molecule, the repressor molecule cannot bind to operator, and thus the synthesis of these three genes takes place. So clearly keep it in mind that when inducer is present, lag operon will be turned on. When inducer is absent, the repressor molecule will bind to operator and lag operon is turned off. So here generally in case of when the inducer is absent, lag operon is in turned off condition. So one by one we take. So the first genotype of a bacteria is such as when the regulator gene. So here the plus sign indicate that this regulator gene is normally functioning. This is a good copy of this gene. So regulator is in good condition. That's why it is represented by by a plus sign. And promoter is in good copy, operator is normally functioning, and this structural gene Z like Z gene is normally functioning and like Y gene is normally functioning. Here we are not representing A gene because it has no more significance. So here this is the genotype of case first. Suppose this is case first, when a normal copy of regulator gene is there, normal copy of promoter, normal copy of operator, normal copy of structural genes, that is lag Z gene and lag Y gene. You know lag Z gene produces beta galactosidase, the enzyme that used to break the lactose molecule into its glucose and galactose. And lag Y gene is the gene that produces enzyme that is required to transport lactose from the outside environment into the bacterial cell. So when the inducer is absent, so what will be the effect of the on the phenotype of these genes? That is, phenotype means the protein product, whether these genes will produce or will not produce. Similarly here, when inducer is present. So as you know, if the inducer is absent, such as if lactose is absent, allolactose is absent, so in that case a normally functioning regulator gene will produce repressor molecule, and these repressor molecule will bind on operator, and then there will be no production of Z and Y gene. So here this in case of the inducer absent, there will be no production of Z gene and no production of Y gene product. So it is represented by minus. When a gene is produces protein, then that is represented by plus, it will come later on. And when the inducer is present, you know this inducer inhibits the repressor molecule or the gene I product to bind over operator, and in that case the RNA polymerase come and bind at the promoter and starts transcription of lag Z and lag Y gene, and if the transcription takes place, the protein will be produced. So when the inducer is present, then like Z gene will be produced, like Y gene product will be produced. So now the case second. Second case is when a normal good copy of regulator gene, good copy of promoter gene, good copy of operator gene, and good copy of lag Z gene, but a defected copy of Y gene. Look carefully. Minus sign represents that this gene is mutated and is now producing a defected protein or is not producing a protein at all. So here what happens if inducer is absent, the regulator gene is normally functioning, will produce repressor molecule that will bind on operator, and hence there will be no production of Z gene and no production of the not expression of this Y gene. So in this case there will be both will be negative, that is no production, no expression of any gene product. But what here, if inducer is present, just like the case in just like in first case, similarly when inducer is present the expression of genes takes place, we generally know it, but here the Z gene is normally functioning, so its product will be available, but the Y gene is defected or mutated, so here its product will be unavailable. So the third case, when the I, that is normal regulator gene, normal promoter gene, normal operator gene, but a defected Z gene and a normal Y gene. So in this case, when inducer is absent, what will happen? If inducer is absent, in all that case, the regulator gene will function dominantly, and here the Z gene is mutated, so already there will be no production of Z gene. However, it is mutated, it still will not produce in any case if the inducer is present also, and the Y gene is normally, but here the inducer is absent, so the repressor molecule will be dominant, so that there is no expression of Z and Y gene. But what in case of inducer, when inducer is present, here we can say that when inducer is present, the I gene remains not more effective, and here the Z gene is defected, so it is not going to produce or going to transcribe, so here the Z gene product will be absent, and here the Y gene is normally available, good copy of Y gene, lag Y gene, so here the product of lag gene will be produced, that is the beta galactosidase permease. And now what is the fourth condition? So we have to represent it from the above side. Now the case is the fourth case. So fourth case, when the good copy of regulator gene, good copy of promoter gene and operator has undergone a mutation that is represented by C, that is constitutive mutation, we will come here to describe it, and Z gene is also in good copy and Y gene is also in good copy. So whenever the mutation takes place on operator sequence, in that case the regulator gene product, that is repressor molecule, cannot bind at operator, and RNA polymerase comes and starts synthesizing these gene product. So what happens when inducer is absent? In that case, when inducer is absent, the normal copy of regulator gene will act normally, and it inhibits the production of leg Z and leg Y gene. So in this case, sorry, when the operator gene is mutated, it means in this case lag gene product is not able to bind on the operator gene. Definitely, if operator sequence is mutated, then regulator gene product remains no more functional to bind on the operator, and in that case, if even the inducer molecule is absent, the RNA polymerase will come to bind at promoter. Even if inducer molecules are absent, and RNA polymerase will bind over promoter, because no one is there to block the binding of RNA polymerase at promoter site, and there will be synthesis of both lag Z and lag Y gene even if the inducer is absent, provided that there is mutation on operator sequence. And this C for constitutive mutation, it means if operator sequence has occurred a mutation, there will be continuous synthesis of these gene product, that is, it is constitutive mutations. And when the inducer is present, further, when inducer is present, if even in absence of inducer both gene product are available, then if inducer is present, it will be further a positive thing. So here we will have the product of these both genes. Now the fifth case is here, when the normal copy of regulator gene, normal copy of, no, here it is mutated copy of operator, and normal copy of operator, and normal copy of Z gene, and normal copy of Y gene. Listen here, and in absence of inducer, look here carefully, that in this time there is mutation on promoter site, and if a promoter site has occurred a mutation, then RNA polymerase cannot bind on promoter, and there will be no transcription of these genes. So when inducer is absent, there is there is no significance of repressor molecule, and repressor molecule will bind on operator sequence, and as a result of which no production of protein. And similarly, if inducer is present, here this inducer molecule has no significance if the promoter sequence is mutated, because RNA polymerase cannot bind, so there will be no expression of these genes. Another case, that is the sixth condition. So when there is IS, that is regulator gene has occurred a mutation, that is S mutation, and normal promoter is there, normal operator is there, normal structural genes are there. So what happens? What does mean by this S? So this is a type of mutation in regulator gene, and this is known as super suppressor mutation. Super suppressor mutation. Super suppressor mutation. That is when this type of mutation occurs on regulator gene. This regulator gene produces the repressor molecule, and this repressor molecule has increased affinity to bind over the operator sequence. Even if the lactose is present or an effector molecule or an inducer molecule is present, these molecule remains no more functional, means they have no more role if the super suppressor mutation takes place in regulator gene. In that case, the regulator gene product, that is suppressor or repressor molecules, these molecules have a strong tendency to bind over operator, and they inhibit the binding of RNA polymerase over promoter. It may be that lactose is present, these suppressor molecule may even form a complex with the lactose molecule, and even in that condition they have the strong ability to bind at the operator sequence. So in either case, whether lactose is present or absent, these genes are not going to transcribe their product. So in this case we are getting no product of any gene, because this is a dominant mutation and a strong mutation, and here this suppressor molecule has increased tendency to bind at operator, and even if lactose is present, these proteins cannot be removed from binding on operator, and hence there will be no transcription of any gene in any condition, whether inducer is present or inducer is absent. And now the seventh condition, is when the regulator gene has normal mutation as represented in other gene cases, normal promoter, normal operator, normal structure genes. Normal is represented by plus and mutation is represented by minus. And here plus and minus represents the production of their enzyme, or minus represents the absence of the enzyme production, plus represents the uh, production of enzyme. So what in this case, when the inducer is absent, so as we can see that mutation has occurred on regulator sequence, unlike the previous type of mutation, that is IS. This is a dominant type of mutation, and in this case the regulator gene product has increased efficiency, increased affinity to bind over operator sequence, and then to turn off the lag operon. But in this is another type of mutation, that when a mutation has taken place on regulator gene, then these regulator gene product, that is suppressor molecule, has reduced efficiency, reduced affinity to bind over the operator, even in presence of inducer or in absence of inducer. So if mutation has taken place on regulator gene, this suppressor molecule will have reduced or no affinity to bind over the operator, and if the inducer is absent, then in that case RNA polymerase will come to bind over promoter and starts transcribing these genes, because here the repressor molecules, those where turning off the lag operon, are already non-functional. So here the expression of both gene takes place, because both genes are in normal copy. And here in case of when inducer is present, this is a further positive thing to turn on the or efficient expression of these regulator gene, these structural gene, and so all genes will have their product. So this is the case, that how the gene regulation takes place simultaneously in different in different combinations of normal and mutated gene. And this was the case for a haploid bacteria, and bacteria are always haploid. But what if a bacterial cell which is haploid, if this bacterial cell can become diploid, or if we make this bacterial cell a diploid cell for particular gene. A diploid cell is that possess at least two copy of one gene. Whether these copy are same or different, these different copies are known as alleles, and same. So this bacteria which is haploid can become diploid for these lac genes. How we know that a bacteria has its chromosomes and another extra chromosomal DNA, that is known as plasmid. And here this plasmid is known as F plasmid, F for fertility plasmid. If this bacterial cell has this plasmid that is outside or other than the bacterial chromosome, that's why it is known as extra chromosomal DNA dispersed in its cytoplasm. And this this bacteria will be known as F plus, that it has the gene, and now it is represented by F plus. And this bacteria, whenever undergoes conjugation, when a conjugation tube formation takes place between two bacterial cells, and there is the transfer of one copy of this plasmid DNA, one copy of this plasmid DNA through this conjugation tube to the receptor bacteria. So here this bacteria receives one copy of this plasmid, bacterial plasmid, and this one copy produces its complementary copy. And now this second bacteria, which was primarily negative, F negative, now after getting one copy of this plasmid becomes F positive. So if we can transfer the lac operon gene on this fertility factor, and then this lac gene is transported to a bacterial cell. Here the bacteria will become diploid for this lac gene. Why? Because its DNA, its chromosome already has one copy of lac operon, but it gets another copy of lac operon from another donor bacteria. So this bacteria can become diploid for lac gene. So what is the case if a bacteria is diploid for lac gene, and how the genes are regulated? So here we are going to discuss the same condition as in case of the haploid bacteria. So here, so the first case, now this bacteria is diploid, that is it has two copy of each gene of a lac operon. It means it has two regulator sequence, two operator sequence, two copies of each structural gene, two promoter sequences. This is, so what if this bacteria has normal one copy of regulator gene, normal promoter, normal operator, normal structural gene of both, and this is one copy of lac gene, and another copy is represented here. Normal regulator gene, normal promoter gene, normal operator, and normal structural gene. So these genes are represented by a slash. It means this set of gene is present on one chromosome, and this set of gene is present on another chromosome. So it is represented by a slash. It means both these set of genes are present on separate DNA molecule or separate chromosome. As we know that one copy is present in bacterial chromosome and one copy in the fertility factor or F plasmid, that is extra chromosomal DNA. Extra means other than the chromosomal DNA. So what happens if the inducer is absent? Inducer is absent, what will be the effect on the product of lag Z, lag Y, a gene, and when the inducer is present, what will be the effect on lag Z and lag Y gene? As we know that this is these two set of genes is now equivalent to the haploid bacteria, because if a haploid bacteria has a